In 2016, a Japanese cell biologist won the Nobel Prize in medicine for proving something strange about your body. Your cells eat themselves, not destructively, as maintenance. When a component inside a cell wears out or gets damaged, the cell wraps it up, breaks it down, and reuses the parts. A recycling system running inside every cell you have right now, constantly. The name for it is autophagy, Greek, literally for self-eating. Here's why that matters to you. That cleanup slows as you age. Damaged parts stop getting cleared, they accumulate. And that accumulation is increasingly linked to the diseases we treat as simply part of getting old. So the obvious question is whether you can switch it back on. The answer involves a compound sitting in ordinary food. It also involves a supplement industry describing the evidence rather generously. Both are in this video. Ohsumi didn't discover that autophagy existed. Researchers had glimpsed it back in the 1960s. The problem was that nobody could work out how it happened, and it stayed a mystery for 30 years. What he did was use ordinary baker's yeast to hunt down the genes controlling it. In 1993, he published 15 of them. Baker's yeast, the same organism in the bread on your kitchen counter. That's what cracked it open. Once you know which genes run the machinery, you can start asking what switches the machinery on. Which brings us to a research group in Austria, led by Frank Madeo at the University of Graz. They spent years working out what reliably triggers autophagy in a living organism rather than in a dish. One answer kept coming back, a compound called spermidine. It isn't synthetic and it isn't rare. It's been sitting in ordinary food for as long as humans have been eating. Legumes, whole grains, mushrooms, aged cheese, which predictably created a commercial opportunity. You can now buy spermidine in a capsule for 30 to 80 pounds a month. Hold that thought. How that story ends is the most interesting part of this video, and it's the part almost nobody covering this topic mentions. So, which foods actually contain it? There are 10 that hold up when you check the underlying data. I'm going to split them into two groups and I'll tell you why. But first, one thing that separates this video from most on the subject. The measured content of any food varies enormously between studies. Wheat germ gets quoted anywhere from 240 to 350 milligrams per kilogram. Soybeans from 167 to nearly 300. Those aren't rounding errors. So anyone handing you a confident ranking, number 1 down to number 20, is giving you false precision. What the data does support is grouping. Here's the first group, the 4 that come out high in every dataset. Wheat germ, the nutrient-dense part of the kernel stripped out when flour is refined. The most concentrated common source anyone has found, and the exact form used in the clinical trial will come to. Stir it into porridge. Soybeans, measured across realistic portions rather than lab concentrations, cooked soybeans top the list. Edamame, tofu, tempeh, natto, fermented soybeans. Fermentation pushes it higher still, and it carries vitamin K2 in the MK7 form, which has its own cardiovascular research behind it. Aged hard cheese. As bacteria break down proteins over months, polyamines get released as a byproduct. That's why a 2-year cheese tests dramatically higher than a fresh one. Notice the pattern. Fermented and aged foods test higher than fresh ones. That isn't marketing, it's chemistry. Below those 4 sits a second group of 6. Reliably moderate, measured repeatedly, and far easier to eat every day. Green peas, lentils, mushrooms, especially shiitake, broccoli and cauliflower, where raw tests higher than boiled because some leaches into the cooking water. Whole grains, measured consistently across UK, Norwegian and French studies. And nuts, with peanuts noticeably ahead of almonds or cashews. That's your 10, and here's what I'd point out about all of them. Every single one is a food a nutritionist would already tell you to eat, for reasons that have nothing to do with spermidine. Fiber, plant protein, polyphenols. Remember that because it matters at the end. Now 4 foods that didn't make that list, and why. These appear on almost every spermidine list online, and the number behind them falls apart when you check it. Citrus. Oranges get put on these lists relentlessly. Look at the analytical data across UK, Japanese and Norwegian studies, and orange comes out modest at best. Nowhere near peas or lentils. What citrus is actually rich in is a different compound called putrescine. Same family, related molecule, not the same thing, and not the one the autophagy research is built on. Liver has has the same problem. It shows up everywhere as a polyamine powerhouse. Liver is genuinely the highest measured source of spermine, again a relative, again not the compound in question. Its actual spermidine content is modest, and durian and amaranth appear constantly. I couldn't trace a confirmed figure for either back to primary research. They're fine foods, I'm just not handing you a number I can't stand behind. If you've seen a video listing 20 foods with a confident figure beside everyone, some of those came from other blog posts rather than from measurement. So that's the food. The bigger question is whether it shows up in how long people actually live. There's a study for that, and it's a good one. Bruneck, a town in northern Italy. Researchers followed 829 adults for 20 years, assessing their diets repeatedly rather than once at the start. Over the follow-up, 341 people died. Sorted by spermidine intake, the pattern was hard to ignore. Lowest third, deaths ran at roughly 40 per 1,000 person years. Middle third, around 24. Highest third, 15. The researchers put the gap between top and bottom as equivalent to being 5.7 years younger. And of 146 nutrients they analyzed, spermidine showed the strongest inverse relationship with mortality of any of them. It was then independently replicated in a separate Austrian study, and a large American analysis of nearly 24,000 people found the same direction. That is a genuinely strong body of observational evidence. Now, two things about that study that I haven't seen a single video mention. Both are in the paper. First, where was the spermidine actually coming from in that Italian cohort? Not natto, not wheat germ. The two biggest contributors were whole grains and apples and pears, ordinary food, which makes the finding less about exotic longevity foods and more about eating a normal plant-rich diet. Second, and this one is genuinely awkward, the people with the highest spermidine intake also had higher glycemic load and higher rates of type 2 diabetes. The researchers reported it and didn't explain it. That's what an observational study looks like when you read past the headline. It can tell you two things move together. It cannot tell you one causes the other. People eating more spermidine were eating more whole grains, more fruit, more vegetables, more legumes. Is it the spermidine or everything else that comes with eating that way? No study here can separate those. Which brings us to the test that actually settles things. Observational studies watch what people already do. A randomized controlled trial takes two matched groups, gives one the real thing and one a placebo, and nobody knows which until the end. That trial exists. It's called SmartAge, run out of Charité University Medicine in Berlin, published in JAMA Network Open in 2022. 12 months, double-blinded, placebo-controlled, older adults who had noticed their memory slipping, exactly the group you'd want to help. Compliance was 95%, so people genuinely took the capsules. They were given a wheat germ extract, the same food that tops the concentration list, and the result was nothing. No improvement in memory over placebo. No improvement in the biomarkers either. The change in the primary outcome was minus 0.02, with a confidence interval running from minus 0.08 to plus 0.04. That interval crosses 0, which is the statistical way of saying we found no effect. Here's the part that made me want to make this video. While researching it, I went looking for how that trial gets described online, and I found supplement retailers describing the SmartAge trial as showing that participants taking spermidine improved their memory compared to placebo. That is the opposite of what the paper reports. Same trial, same journal, same year, described as a success by websites that earn commission when you buy the capsules. I'm not naming them because it isn't one company, it's a pattern. When the evidence doesn't say what a product needs it to say, the evidence gets quietly rewritten. So if you take one thing from this video, make it this. When you see a health claim attached to a study, check who benefits if you believe it. So where does that leave you? The Nobel Prize discovery is pseudoscience. Autophagy is real, and Ohsumi mapped the machinery. Spermidine triggering autophagy and extending lifespan is strongly established in yeast, flies, worms, and mice, proved genetically by knocking out the autophagy gene and watching the benefit vanish. In humans, dietary intake tracks with lower mortality across multiple large populations, replicated independently. Encouraging but observational. And the one rigorous trial of supplementation found nothing. So here's what I'd do with that. Look at what those 10 foods have in common. Everyone has its own independent evidence base. Fiber, plant protein, polyphenols, vitamin K2. You don't need the spermidine story to be true to justify eating them. That's the point. They're a good decision whether or not this molecule turns out to be the reason. What you don't need is the capsule. A tablespoon of wheat germ costs pennies and contains more than most supplements deliver. If you want more of this, the version where the studies that disagree get mentioned too, subscribe. That's what this channel does.